So you have seen already several talks about catalysts and catalysis in this global XAS seminar. That's because quite a few of synchrotron or XAS users are dealing with catalysis. And that's simply because 90% of all chemicals, so every product of chemical industry, or 90% of them that you have around you, they have seen a catalyst during their production or life cycle, or maybe recycling. So we inevitably need to work with catalysts. And the most of people, if you're asked about catalysts or catalysis, they would think about automotive catalysis, so a catalyst in your car, and that's about solid gas reactions. So they're most common and they're relatively easy or relatively stable established tool for synchrotron research. You do that in quartz capillaries most often, and that works, and many people already know how to do that, and you have the infrastructure at synchrotrons. Therefore, I decided not to go for this topic, but I decided to show you something different. So it's not always in the gas phase that you have your reactants or products. Sometimes it's liquid, and sometimes you have to go for higher pressures. An example is hydrogen peroxide. Why is it so important? Because people use it everywhere as bleaching agent in the paper industry or for textile industry, for wastewater or drinking water treatment in mining or metallurgy, as a disinfectant, detergent, in electronic industry. Now everybody talks about the shortage of semiconductors, and that's one of the elements which is required, or one of the bulk commodity chemicals which is required is hydrogen peroxide. And of course, more interesting for me, it's used everywhere in chemical synthesis. Now the problem comes if we look at how hydrogen peroxide is currently produced. You need, or people use, an so-called anthraquinone auto-oxidation process that involves both reduction and oxidation cycle of the anthraquinone. But then if you show something like that, especially the waste from that process to a green piece, they won't be happy. And another problem, maybe a bigger problem for all of us, is that this process is economically viable only at a large scale. So there are only a certain number, not so many plants across the globe which produce hydrogen peroxide when it's concentrated and when it's transported to the end user. And you will be probably surprised if you try to Google, say, train, say, accident and hydrogen peroxide, by how many hits you will find in the Google. So it's really dangerous commodity chemical if we talk about transport. So we need an alternative. An alternative would be a way to synthesize hydrogen peroxide at a small scale directly at the point of use, for example, in containers like that. That works if you just mix hydrogen and oxygen, dissolve them in a liquid phase, and pass over the catalyst. So you can get hydrogen peroxide indeed. Now there are two problems with that. First, when we are talking about dissolving hydrogen and oxygen in liquids, we need high pressures, so high partial pressure to get high concentration of a dissolved gas in a liquid. And second, hydrogen peroxide is an intermediate product, and the final product in the chain of transformations is water, inevitably. And there is another problem. Hydrogen and oxygen can react directly without making hydrogen peroxide, but instead, if we're talking about gaseous reactants, making an explosion. That's definitely not what we want to do. So we need to solve all those problems. As chemists, we need to solve the problem of selectivity. That means we need to find the catalyst, or more precisely, the state of a catalyst which would be selective to making hydrogen peroxide and not water. And then as engineers, some of us in the team were engineers or are engineers, we need to solve the problem of not exploding ourselves and the synchrotron during our research, so the safety problem. And what we decided to do previously, how it was done, so there were a couple of publications on the studies of catalysts for hydrogen peroxide production. But when to make it safe, what people did is that they took hydrogen and oxygen and diluted it by 20 times, just simply to go out of the explosive regime. So it's not really working because, as I told, we need high partial pressures. So we cannot simply dilute. In this case, we do not dissolve anything in the liquid. So it's a catalyst with a solvent, but not with hydrogen or oxygen, not producing anything. Instead, we decided to do as follows. So if we cannot mix hydrogen and oxygen in a gas phase, why not doing it in a liquid phase? We anyway need to dissolve them. So we made a setup with 2 saturation units for the hydrogen in a liquid in a separate container, and then oxygen in a liquid in a separate container. And when pumping liquids together, mixing them, so we have a mixture of water. For the first experiment, we had water as the most harmless solvent. And then with hydrogen with relatively high concentration and oxygen with relatively high concentration. Then a reactor, which we are free to choose what we want. Of course, we want something on which we can put on a beamline and measure spectra over that. And then analytics, that was titration in our case. So if we are talking about the in-situ cell of a reactor, as I told, we are free to choose. And so why not go to something tailor-made for this specific project? Of course, if we require some expensive machinery, that's maybe difficult for everybody. But if we're dealing with Teflon PTFE, that's a brick of PTFE costs you $5 on eBay, and you can machine it in your garage. So it's quite simple. And compared to quartz capillaries, you can tune it directly to the size or volume, or I'm talking about beam path length, so that you get good spectra, not simply what you have off the shelf, but spend a couple of hours in your garage and size it to your needs. As X-ray windows, we're talking about palladium catalysts, so quite high energies. And therefore we took polycarbonate. I mean, it's not something what you read in the textbooks when you are going to read about windows for X-ray absorption spectroscopy, but it works in our case for 24 keV, so why not? And we get an advantage that we see what happens with our catalyst bed. Since we are not dealing with gases, but with liquids, and that means we have high viscosity, high pressures, we expect to have movement in the catalyst bed. That's not what we want to have for X-ray absorption studies. Of course, we want highly homogeneous samples for transmission. But let's face the facts. If we want to run it on real reactors, we have to live with compromises. So at least good we can see what's happening, also with a camera. Now that's how the first setup design looked like at DAISY P65. That was already a few years ago. And I remember the first question from a safety manager when he came in and looked at that. So here is your bomb. Yeah, those are pressurized cylinders with oxygen, pure oxygen, and pure hydrogen. Only 10 bars, so not too high, but still or not completely safe. Or I mean, it's safe with all stainless steel, but I imagine safety responsible people would think about that twice. With micro gear pumps and then a cell in the beam, so transmission measurements. Back pressure regulators somewhere not visible here, and analytics under the table. The procedure, of course, we need to design the experiment in such a way that we can measure as much as we can, both with respect to catalytic data and spectroscopy. So And of course, we always need references. Therefore, we went for the XAS sample. We want to know how the XAS sample looks like. But more interesting is what are the differences between XAS and Insitu. Many people measure XAS, but it's not always the same. Just to confirm that, we need an XAS reference. And then Insitu references, reference in oxygen-saturated liquid, so oxygen-saturated water, and hydrogen-saturated solution. And the Operando or Insitu data. So since we did not know exactly in which regime we are going to have the highest production of the hydrogen peroxide, we went for different residence times, so different flow rates relative to the amount of a catalyst in, and different hydrogen to oxygen ratios. Most of the pre-studies, also in literature, are done in the batch. We are not working with the batch reactors, we are working with the continuous reactors, as also industry prefers to do. So therefore, we cannot really get from the literature exact conditions. We need to improvise, and ideally we need different conditions with different reactivity and different selectivity. So to build the structure-activity relationships, it's not enough just to find the condition under which the catalyst is active. We need always to have a comparison. So we need always good and bad references. And what we found out is that for some of the conditions, we didn't have hydrogen peroxide with too much hydrogen because simply it was overreacted to water. Then at too low hydrogen to oxygen ratio, there was no reaction at all. And then around the stoichiometry, around 1:1 hydrogen to oxygen ratio, we got quantifiable amounts of hydrogen peroxide. The spectra looked roughly the same, so I will show only one in this part of the talk. And let's have a look at it. No matter be it reference conditions, water, which is by default saturated with oxygen, or hydrogen saturated water, so reducing conditions, or hydrogen peroxide synthesis conditions, the spectra looked roughly the same. I must note that the ex-situ catalyst shows palladium oxide, so in-situ catalyst shows more or less metallic palladium features. And the spectra were measured, as I told before, we had 6 conditions at least, and then for each conditions, we measured at different points in the catalyst bed, at the inlet, at the outlet. When you are studying catalysts, that's also important to remember that in plug flow reactors, you have a conversion, which means for fast reactions, you may have at the inlet of a catalyst bed, the catalyst which sees only reactants. And at the outlet, it may happen that there are no reactants at all left, and the catalyst sees only products. So it's quite often the case that if you try to measure somewhere in the middle, or at the inlet, or at the outlet, you will see different spectra. And simply because of the kinetics, you have different environment, depending on where you measure them. So you need to measure ideally at 2 points, always. And also purely for statistical purposes. So from the first look at the XAS, it's roughly already we have guesses on what's going on. With too much hydrogen, we have lattice expansion in the XAS, so palladium-palladium interatomic distances are shifting to higher values. And then with too much oxygen, or also while with a working catalyst, that's the red curve in the middle, we also observe palladium-oxygen feature, so the oxygen light element in the first coordination shell. Then if we compare it to reference data, and it's important to have reference databases, that's a big project in Germany right now going on, to establish reference databases available to everybody, which would be searchable and with reliable data in it, with proven data modeled and with X-ray diffraction data to support the reference data. So if one has such a database, one can already observe the features also in the xanes, like the high white line due to oxidation of palladium, and the shift is related to formation of beta palladium hydrides. So there are two kinds of hydrides. If we are talking about palladium alpha hydride and beta hydride, the difference is in the alpha hydride, we do not have the lattice expansion of palladium. So X is within or below 0.3, and above 0.3, we have a bulk hydride formation with lattice expansion. So for our catalyst, of course, it's difficult to get a beta hydride reference. We need to do some in situ measurements, ideally with large nanoparticles if we want data on the bulk hydride, but we didn't have that. We had only our catalyst, and it appears that it's enough just to have 10 bar of hydrogen saturated water to drive it fully to the beta hydride regime. So we had those reference, and with the help of the references, we could quantify both. We could quantify the oxidation state, and for that we used both xanes analysis, linear combination. Actually, no, I think, yeah, that was some time ago. I think we used simply the intensity at this energy point and compared to the references. And here is for the numerous data sets, the data, and then the hydrides, so hydrogen saturated water reference and oxygen saturated water reference. And both we used also coordination number from the XAS analysis. Both gave us roughly 10 to 15% of PDO fraction, which actually corresponds to fully covered surface of 2 to 3 nanometer nanoparticles. That's the TEM data. So XAS and xanes analysis tell us that in the working state, so experiments 2 to 6, most of them producing hydrogen peroxide here. In the working state, we have palladium nanoparticles with surface covered by chemisorped oxygen. Now, if we look at the features of palladium hydride, it's again a question of how to quantify them, because the features and the shifts are rather small, so we cannot simply use the standard linear combination analysis. We need to find something else. And we can use in the xanes shifts of the features. So the peak B is actually a peak from the XAS already. Also the shift of the H, and also how it was suggested by Bugaev et al. That one can use the ratio of intensity of A over B peaks. So both ratio of A over B peaks intensity again lies in between the red hydride reference and black oxygen reference. So we are somewhere in between, but not the beta hydride. And with respect to the rising edge position, we are also somewhere in between. Unfortunately, I can't, something in between the red hydride reference and the black oxygen in water reference. So we have virtually no palladium lattice expansion that we get from XAS data. We have that's valid for the green and yellow marked data. So we have high disorder for those data sets, which is sometimes a signature of hydrogen disturbing the lattice. So the electronic structure corresponds to palladium hydride, but the changes are not so strong as we see them in beta hydride. And that is typically attributed to alpha hydride, so no lattice expansion in the working catalyst. With that, I'm nearing the conclusion about the first part of our work. So we got an information about which kind of nanoparticles are actually active to produce hydrogen peroxide in water. So palladium nanoparticles with chemisorbed oxygen on the surface, and not too much hydrogen in the lattice, so without lattice expansion, with X below 0.3. That corresponds to alpha palladium hydride. This state of catalyst produced indeed the highest amount of hydrogen peroxide. And to confirm, we had an Operando experiment. We measured the concentration of the hydrogen peroxide, which we obtained from that experiment, and compared it against the data measured in the lab. So light green here are the data from the lab measured with the Insitu cell as a reactor. And then we have black and blue data that's with a stainless steel tube as a reactor. As you can see, in the Insitu cell, we are losing quite a lot of productivity, and that is due to PTFE. As I told, it's quick and easy and inexpensive to use Teflon as a cell body, but we are losing hydrogen. It just simply diffuses through even thick Teflon cell body, and it diffuses through polycarbonate X-ray windows. But still we have enough to be able to measure on a working catalyst. And then we did some homework about which I will report later, and repeated the same study on the same catalyst in alcohols. Why alcohols? Because for chemical industry, they need solutions of hydrogen peroxide in alcohols. Ideally, we would like to work with methanol, but we do not want too much methanol at the beamline. So we went for ethanol based on the fact that the chemistry is essentially the same. And we again found out that the most active and selective to hydrogen peroxide catalyst had not a beta hydride state of the nanoparticles, but a mixture of alpha and beta, closer to alpha, so very slight lattice expansion. And the difference between ethanol and water is that ethanol effectively cleans the surface of nanoparticles from the chemisorbed oxygen. Again, if you dose too much hydrogen, you lose hydrogen peroxide production. It produces water, so it's unselective. At that point, I can wrap up again. On one hand, we got an Operando data and we know how the active catalyst looks like. But on the other hand, we had a lot of problems. So only semi-quantitative production of hydrogen peroxide, very low yields, hydrogen going out through the cell, instability of the catalyst bed, and unstable analytics due to bubbles distorting the analysis. But even with all those problems, it was better when people could do it before. So the experimental setup was called ingenious, but an expert doubted that it could be scaled up. So at that point, I will stop for questions and we will continue with scaling it up. So,
which is the proof that you should never listen to experts when they criticize your work that you're happy with.
No,
very
take
good.
It as a challenge.
Very good. All right, Yang Ha, you had a question?
Yeah, so it's a very interesting study. So I wonder if you're are you monitoring the surface of your catalyst, or are you looking at the overall signal?
No, we are looking at the transmission through the whole cell, through the whole catalyst.
Okay, so it's a transmission mode. So you are looking at basically a signal from everything, which means
yes.
If you don't see too much difference in among different cells, it might be because the catalytic reaction only happens at the surface while you're looking at the overall signal. So is it possible to evaluate like the amount of signal from the surface based on the size of your nanoparticles?
Yes, and that's what we did to evaluate the oxygen coverage. So we had the amount of a change, basically, of a fraction of the oxidized palladium, and then we related it to the size of the nanoparticles, and it corresponded very well.
Okay.
So yes, unless the nanoparticles are very large, in which case you will not see the surface effects at all. So that's a potential problem with catalysis in general, you need to make sure that your fraction of the active material is large enough to see that. So in general, it depends on your material, but for these materials, it is possible, yes.
Cool, thanks.
Anatoly, did you have an independent question or something you wanted to add on to that?
Oh, just wanted to add on to it.
Okay, go ahead.
I'm not the next one because I think Hudson was before me, but anyway, since you brought it up. Yeah, Dmitry, an interesting possibility to determine the size while you do these experiments is by using hydrogen hydride formation as the probe, because the type of shifts and the corresponding occupancy of hydrogen scales up with the particle size. We showed that in 2013, it was 1 year before Bugayev's paper, but it's just a cool method of determining the particle size while you do this type of work.
I actually think we referred to that paper of yours, and we definitely had that discussion with referees of the first paper, simply because indeed coordination numbers are changing. Even that you can see dependent reversibly, depending whether you go to a hydride or decomposing with hydride. But yeah, the system is dynamic, it's for sure.
Thank you.
Hudson, you have a question?
Yeah, thank you. Thanks, Dima. I was wondering, because you have a kind of a large reactor, yeah? And as you said, it's not homogeneous, yeah? Chemical species at the beginning, thinking of the catalyst, are of one kind, and at the end they are of other kind. And I have the feeling that synchrotrons are going towards even smaller and smaller and smaller beams. And how you deal with this problem, because you are looking to such a heterogeneous system, and how representative is it from the whole catalyst?
Yeah, so that's a good question. And unfortunately, as a chemist or as a person who doesn't want to burn catalysts, I can say, unfortunately, some people see differently. But unfortunately, synchrotrons are going towards smaller beams. I can tell, if you mean representability, repeatability, it's still quite good. So on one hand, we never measure, or let's say, if we can get more than 1 point within the cell for the given beam size, when we are measuring at different points. So we are measuring more than one spectrum. And then in the worst case, you need to model also kinetics to relate this single spectrum at the given position of a catalyst bed to the modeled predicted concentration of the incoming gases and the products. That's actually what Jeroen van Bokhoven did for some of those studies. We also did kinetic models. But else, if we come back to the same point where we measured before, it's reproducible, so it's not a problem. Unless we're dealing with rigs or the data which require focusing of the beam, in that case, it may be a problem. So if we cannot keep the precise focus to, let's say, the precise same catalyst nanoparticle or precise distance, then it's sometimes a problem. Like for platinum, for very sensitive measurements, for platinum L3H oxidation state, we had Riggs measurements. They definitely depended, even for platinum oxide reference, it depended on plus minus a millimeter of focal distance. That's a problem, and for that, I do not have a proper solution. But like conventional XAS, yeah, if you go back to the same point in the catalyst bed, then it's reproducible.
Because what you try to do is to combine the yield, yeah, the catalytic yield, and with the chemical species. This is what you are trying to do, to find which is the active chemical species of the catalyst. But the chemical composition of the catalyst is changing along the bed.
Yes. as the gas composition is changing. So what you need to know in the end is precisely what's the gas composition, and when you get precise information about what the catalyst composition. And just remember that it's changing indeed, yeah. But both you can get, be it measuring in a specially resolved way or modeling of a microkinetic modeling.
So you would prefer a larger beam or a smaller beam?
It depends. I would prefer a beam which is spanning across a few grains, not too small beam, but it depends. I mean, if it's, yeah.
Okay, last question for this break. Janos, you have a question?
Yeah, I didn't catch, Dmitry, what was the particle size of your palladium nanoparticles, and also how the reactivity was changing if you studied with the particle size of the palladium? Because if you go down to very small palladium particle sizes, I assume that your surface species are changing with the particle size as you go down to very low palladium particles.
It's not only the surface species, but you are changing also the hydrides, also the kinetics of the hydride formation dramatically.
Yes, exactly.
So let's say, in short, that is mostly not studied, or definitely not studied in a consistent way. A bit longer, the smaller the particles are, the less they are active, and also the lower probability of forming any hydride. So ideally you go for not too large particles, and let's say 5 nanometer particles work better, and that's what I will show. I mean, much better. That's what I will show later. And probably we can take that discussion. It's a very interesting discussion, but to the end of the talk.
Thank you.
Yeah.
Very good. Dr. Doronkin, you should continue, please.
Yes, sorry, the discussion is nice, I think. Let's continue now. What can we achieve if we do reasonable system optimization? And the first thing would be to check for better catalysts, because the previously used catalyst was what was prepared by engineers just to test the setup. And then what can we do with the setup and the Insitu cell to make it work better? So let's go and start first with the Insitu cell. As I told, Teflon you can machine in a garage, so that works nicely, and that allows you to get nice results sometimes, but you get much better results with a proper stainless steel equipment, which is tested for high pressures. And when engineers calculated also permeation of hydrogen, permeation of oxygen, stability of window materials, O-rings, and all this, all the process of the calculations for this given Insitu cell that's described in this paper. Benedict Deschner, who did this work, mostly he's an engineer, and I quite like his approach, because all of that was tested also theoretically, calculated. So we had higher pressures, we have negligible hydrogen permeation, we have now metal filters to keep the catalyst in place, so no matter how the viscous liquid pushes it, it can't go anywhere. So we have now a better cell, and that's how it looked at the beamline. And now what about scaling up the setup? So the previous setup was not truly continuous. Now they came with the same approach, mixing two liquids, we've saturated with hydrogen and oxygen, but this time we have a continuous supply of liquids from the containers with HPLC pumps at high pressure, and then continuous supply of pure oxygen and pure hydrogen with MFCs. They are dissolved each in the individual stream of a liquid solvent, so in gas absorbers, which are simply stainless steel tubes packed with silicon carbide to help mixing the liquid and the gas. going through the Insitu cell, so now it's the same, and then depressurizing back pressure valve and going in the liquid product container. Previously, we relied on automated photometric titration as an analytics to make it online. We decided that it doesn't pay off. So what we did now is that we started flushing the liquid product container, and that is to remove unreacted oxygen and hydrogen, not to make an explosive atmosphere in this container. And then it's flushed directly in the mass spectrometer and the rest to the exhaust. So that's an indirect measure on how much gases reacted, but that can allow you to calculate the production of hydrogen peroxide in a real time with an error within 20%. So after the experiment, we actually took the liquid samples and analyzed them, so titrated them in the lab and confirmed that the mass spectrometry actually works quite nicely as an indirect analysis tool. So we went for the measurements at 80 bar. Now it's roughly 2 orders of magnitude higher pressure, which now allows to dissolve more hydrogen and more oxygen compared to the previous experiment. What's more important, now we have stainless steel, so hydrogen, all hydrogen stays there. It doesn't just go through the cell to the environment. Quite low residence times. This is a very flexible setup, and it allows us to vary the reaction parameters in the very wide range. The reaction medium was basically what chemical industry wants us to use, ethanol and then sulfuric acid as a promoter. Fixed bed plug flow reactor, which the cell is 25 milligram of catalyst, 3 milliliter per minute liquid flow. I must say, for very long experiments like we had, in total 3 experiments took 1 week, we used like 70 liters of ethanol, which is a bit more than the normal user at the beamline uses. But that's how it is, we have to live with that. And as to the catalyst development, we went to a bit higher loading of catalysts, which was synthesized by Sheng Wang in a neighboring group. And they had roughly 5 nanometer palladium nanoparticles, which is now larger than the previous test run. So they were monometallic palladium nanoparticles as a reference, and then bimetallic palladium tin and palladium indium catalysts as the most active catalysts as they were found out in the batch experiments. So now let's have a look at how do they perform. We measured up to, let's say, 80 millimole per liter concentrations in the reaction product solution, which is, if you remember, in the previous experiment, we had a maximum of 0.08 millimole per liter. So now we are 2 orders of magnitude, actually 3 orders of magnitude larger than we were in the first measurement. At this point, we're obtaining hydrogen peroxide solutions which are just 1 order of magnitude below the requirements of industry. And that's without any optimization. That's in the Insitu cell, which we made for synchrotron measurements. So in principle, we can continue with optimizing this system. And then if we look at yields and selectivities, one can note that the highest values lie around 0.5, so 50% yield at 50% selectivity, which effectively means that we have failed to get the kinetics, simply because we get full conversion, and that means we are not supplying enough hydrogen or oxygen. It reacts faster than we can supply it. In this case, it means that the kinetic data is not very usable, but it represents only the lower limit of actual productivity of our catalysts. And that lower limit is actually quite high, so it's among the highest published. So from a chemical point of view, we are happy. Now let's look at the spectra. And the spectra, we got quite nice ones. I'm showing palladium KH spectra. But as you can imagine, tin KH and indium KH are quite close to palladium. So without changing the optics, we also could obtain the tin KH spectra and indium KH spectra. And one can see there are just two groups of the spectra. So that is metallic palladium and a similar spectra with a shift, which we tentatively attribute to beta palladium hydride. The difference between monometallic and bimetallic samples is that the shifts are not so obvious, not so pronounced for the bimetallic samples. So from the XANEs, we see shifts and we can attribute them, the shifts between metallic structure and the hydride. We can see they're less pronounced for the bimetallics, and XAS confirms that. Now we can quantify that. We quantified also shifts in the XANEs, but I'm not showing that in the interest of time. I'm showing just the interatomic distances between palladium and the second metal, so palladium or indium or tin, because XAS cannot distinguish between them. So for the monometallic palladium, we have quite noticeable lattice expansion. For the bimetallics, as we saw it in the XANEs, the lattice expansion is not so high. So now, why is it happening? If we look now at, for example, indium KH spectra, and we can also analyze the XAS spectra in this case, and we do not see any lattice expansion in the local environment around the indium. So around palladium, we observe a lattice expansion, around indium not. We already have a guess what happens. We simply do not get hydrogen in between indium and palladium. That's a guess. But when we ask the DFT group to make calculations for us to calculate the hydrogen absorption energy for the monometallic nanoparticles or model nanoparticles and for the bimetallics, and it confirms that for the bimetallics, so as an input, we used metal ratio the same as we got from the XAS analysis. And for the bimetallics, they confirmed we cannot dissolve so much hydrogen in the structure. It's simply thermodynamically unstable. And for those structures, we also calculated interatomic distances, so it fits quite well. And with those calculations, we could confirm also the structural data. For the excess hydrogen, we have beta hydrides, be it monometallic palladium hydride or palladium indium hydride, with one exception that for the bimetallics, we are not getting hydride ions in the interstitial sites between indium and palladium. So for the very active state metallic structure, we anyway have only low amount of hydrogen. Maybe it's only surface hydride. So if we now look at the data which we recorded at the second metal edge, be it tin KH or indium KH, we analyzed for tin XANEs. We did linear combination analysis, and we could see we have roughly 70% of tin as tin 4+, the rest was metallic tin, tin 0. And when for indium, we couldn't analyze XANEs because XANEs of metallic indium is different from the XANEs of the indium palladium alloy. So we had to analyze XAS in this case, and we could see that indium is also a mixture of oxidized and reduced indium. And in the indium case, we can see quite a clear reduction and then oxidation if we change the hydrogen to oxygen ratio. So for tin, it's mostly stayed oxidized. The reduction wasn't at maximum 5%. So we have a mixture of tin and indium. Tin and indium are a mixture of oxides and the metallic state, and when palladium is fully metallic. Now, this part, to finalize it, I can go and show the structure-activity relationships. That's our ultimate goal, to get the idea of what's the active and stable state of each catalyst. So we need to know if it's active, and for palladium we can see under reducing conditions, 3:1 ratio, it's quite active. So that's the yield shown here, and that's the state of beta palladium hydride. And so it is active with oxidizing conditions, even high excess of oxygen, and that's the metallic state. So for palladium, both metallic and hydride state are reasonably active. For palladium tin and for palladium indium, only stoichiometric and oxidizing environments give you high activity of those catalysts. Especially for palladium tin, you need an oxidizing environment. So what we know is that we have a shift between metallic and the hydride structures. Depending on the chemistry, they can be active in both oxidizing or reducing environments. So we have different activity windows. And now I would like to finalize that part with something that many people do not actually analyze, which is the raw data or quantification of the species in the beam or in the catalyst. Again, the problem with liquid-solid reactions often is that you have leaching, which means the active methyl is dissolved in the liquid phase and leaves the catalyst. If you are doing batch reactions, that is just a stirred tank reactor, the liquid never leaves the reaction zone, so you often do not see that. But in case of continuous experiments, the liquid is always new and freshly coming to the catalyst, and the liquid with dissolved active species is leaving the catalyst zone or the catalyst bed and leaving the cell. So if we do not normalize the spectra, but look at the non-normalized data, we can see we have total absorbance going down, and that's due to the mechanical movement of the catalyst bed, so restructuring of the catalyst bed. And at the same time, we see that the absorption edge, so the edge step is decreasing. Of course, simply by the decrease of absorption edge, it contains both the mechanical movement of the catalyst bed and the leaching, so dissolution of palladium. So as such, by itself, it's not usable, but if you now normalize it per total absorption of the catalyst bed and correct for the absorption by the X-ray windows, now you can quantify amount of palladium and also amount of a second metal. So as you can see, for palladium indium, it's not very stable. Now let's have a look at the stability of a catalyst. And that I should note is not really possible without X-rays. So you cannot really put any probe in a reactor otherwise, which shows you how much metal you have there, unless that metal is colored and you have UVVs, for example. In that case, you can maybe see that, but else you need X-rays for that. And so for palladium, we remember it was active with hydrogen to oxygen ratio 3:1, and so that's the reducing conditions, and it's also stable under those reducing conditions. So we have a stable and active window for pure palladium catalyst. And when we start losing palladium, if we go towards oxidizing conditions, so it's not stable. It may be active, but if we lose half of the palladium in 1 day or in 10 hours, nobody is going to use that process. So we need to find something else. With respect to palladium tin bimetallics, it's rather stable at the palladium edge. So we are not losing palladium also at the point where we have the highest activity of that catalyst in the oxidizing environment. And we are not losing tin either. So that catalyst is active and stable for the hydrogen to oxygen ratio 1:3. So if we need to run our reaction in the oxidizing environment, then palladium tin is your catalyst of choice based on both activity and stability. Palladium indium is unfortunately not stable in in the regime where it is active, that is oxidizing environment. Also, indium is not stable. If we look at the relative concentration of palladium relative to indium, indium leaches first, and then we don't have any more indium to leach out, so palladium catches up. But as an end result, we are losing the precious metal here. So it's just unstable. Why are those differences in stability of palladium tin and palladium indium, or monometallic palladium? We did high resolution TEM also after the experiments, and we found out that for the palladium tin, as you remember, we had a mixture of tin dioxide and tin alloyed with palladium. And the tin dioxide actually made an overlayer. Probably it's porous because still the palladium tin nanoparticles are active, so we have a transport of reactants to those nanoparticles. And we think that this overlayer is actually provided an interface anchoring those nanoparticles on the titanium support. And in case of palladium indium, we do not have an overlayer. More than that, indium leaches out first. So even if indium 203 oxide is our interface layer, it just leaches out first, which means our palladium indium nanoparticles are not anchored anymore on the titanium, so they also disappear from the system. So with this, I conclude this part of the study with some little homework, especially working on the cell, so updating the cell, which this time required a proper workshop, but still is possible with just buy-in pieces. So we got high productivities of hydrogen peroxide formation. We defined the activity window for different chemistries. We defined the stability window, and we found out what is causing the leaching. And of course, we again could identify the structures of active and selective and not selective components. So at this point, I will stop for questions.
Given the time, you've got about 10 minutes left. I'm going to say we hold the questions until after your conclusions.
Okay, I have like 3-4 slides with information more, and so we go next. So where do we go next? As I told, proper kinetics we couldn't measure, so we failed miserably with proper Operando data, but still what we got is the highest published, so it's quite okay for a start. And when we got fundamental understanding of hydride structure, but now let's go for the kinetics. Can we actually get kinetics of formation and decomposition of mono and bimetallic hydride particles? So for going to the kinetics, we decided to skip the real conditions, so not to go for the liquid-solid interface, simply because in this case we have to account for mass transfer in the liquid, which is becoming difficult and takes time. So we went for simple experimental setup with switching between pure hydrogen and pure helium. And more importantly, we didn't limit ourselves with XAS, but we decided to add X-ray diffraction. That is because XAS is sensitive to only bulk, or to everything. It's bulk and surface averaging technique. But X-ray diffraction sees only bulk hydrides with lattice expansion, so only beta hydrides. Of course, if we want kinetics for this fast reaction, we need sub-second time resolution, and we need to go for in-situ measurements. Luckily, with respect to the sub-second time resolution, we have a nice collaboration with a group from University of Wuppertal, who provided the quick XAS and quick XRD setup at the P64 beamline in Hamburg. Both can be measured, both XAS and XRD can be measured with sub-second time resolution. We have triggers, and we can record also metadata with the quick XAS data, so we know exactly which gas passes through the cell at what time. and exactly when did it start. So first, I'm showing the X-ray diffraction patterns. Since we are not dealing with a model catalyst with just palladium nanoparticles, but it's a mixture of titanium, different polymorphs of titanium in this case as a support. So it's industrially relevant P25 support. That means we had quite some, or we had to make the efforts to find the reflections which we can use to identify hydrides and non-hydride nanoparticles. And we found out that we could use, in case of hydride, 311 reflection, and for the non-hydride form, it was hidden under the titanium dioxide reflection. But for the non-hydride, we could use the 222 reflection. So we confirmed the hydride and metallic structure using XAS. And now the last two slides with the actual data. So XAN spectra I'm not showing. We did straight away the linear combination analysis on walls, and we could manage to get reasonable data with 2 spectra per second. Unfortunately, the formation of palladium hydrides is still too fast. So we have only 3 points to fit the kinetic data. That's simply not enough. Also, if we look at the diffractograms, we see pretty much after 1 second, or already at 2 seconds, we see the formation of a 311 reflection of a hydride and the 222 reflection of a metallic structure disappears. So no kinetics possible here and no conclusions, unfortunately. But if we look at the decomposition with quick XAS and quick X-ray diffraction, then we can get kinetics, which we can then feed with exponential decay curves. We can see there are not 2, but 3 structures that we need to take into account. We have an intermediate. So first, the decomposition of beta hydride is relatively slow, after which we have an inflection point and it goes faster. From the XAS, we can just tell that we have 3 components, but we don't know what the 3 components are. But it makes now more interesting to look at the X-ray diffraction data. And with X-ray diffraction data, we can clearly see we have a 311 reflection of beta palladium hydride, which moves to theta angles, which means structure contraction. But it stops at some point. So while it moved, it just breathed out hydrogen, but still we had a hydride structure untouched. And when it stops, so it stepwise decomposes into a metallic structure. And in a stepwise manner, we get the 222 structure. So we can now correlate the XAS data with the X-ray diffraction data and tell that the first step is the lattice contraction of the 311 hydride, which then in a stepwise manner goes and forms the metallic structure. So we did that on monometallic and bimetallic samples. The data is still being analyzed, and we hope to relate it to the kinetics of the hydrogen peroxide synthesis. With that, my last slide is to acknowledge people who helped greatly, and that's a joint work of engineer and calculation teams and synthesis teams and the beamlines. So thank you all, and thanks to the people who contributed, and thanks for listening. Now we go for questions.
Thank you. It's a wonderful talk. We'll start with Janos. You have a question?
Yeah. So Dmitry, you focused all of your study on the palladium phase. You have an active support TiO2. So did you consider that TiO2 may play any role in this reaction, or the influences your palladium species, what you are observing?
So in principle, it may, I mean, TiO2 indeed is known for strong metal to support interactions, but for that you need to reduce it. And let's say in a classical or in the field of automotive catalysis, that means going to high temperatures. This was all room temperature. And let's say like that, in the pre-studies, many different supports were compared, and there was no reasonable evidence to say that there was a support effect, except the acidity of the support. So acid is a promoter for this reaction, and the support was actually treated with acid. Except from that, there was no effect found for the support.
Okay, thank you.
Okay, question from Beatrice.
Yes, the maximum temperature of a cell is 100 degrees C. That's a good question, and that's actually what is written in the paper. So this cell exactly is not heated, and for this specific reaction, we do not need heating, we would rather may need cooling. In general, for heating, you would need to install a heating cartridge somewhere next to the catalyst, which for this cell, there is no space for it. So it would need to be machined. 100 degrees C, it's based on the calculations for the what the windows and the O-rings can stand. So how you can do it without redesigning everything. But how to heat it, one would need to improvise if one takes directly this cell drawings, or you need to make space for the heating cartridge. And the limiting factor for this maximum temperature, again, the mechanical stability of peak at 100 bar was calculated, and it starts basically flowing out. So it becomes too soft to hold 100 bar above 100 degrees C.
Okay, seeing no further questions, thank you very much for your seminar.